Chapter 10, Muscle Tissue. Characteristics of muscle tissue are the following. They all have specialized cells, which are going to be long cells that have myofilaments within them. They exhibit excitability, which means they can receive and respond to stimuli. Contractibility, which means they can get smaller in size and respond with the next characteristic, extensibility, where they can stretch in size, and then elasticity, where they can recoil after the stretch back to its original size. The three different types of muscle tissues are skeletal, cardiac, and smooth. Your skeletal muscles are made up of skeletal muscle tissue, connective tissue, and the nerves and blood vessels of the tissue. Each single cell is called a muscle fiber, and the skeletal muscles are directly or indirectly attached to the bone. There are 5 functions for the skeletal muscle. 1, they produce skeletal movement by pulling on the tendon that pulls on the bone. 2, they maintain your posture and your body position while you're sitting and standing, and your head position. 3, they support soft tissue, such as the abdominal wall, the floor of the pelvic cavity, to support all the internal organs. 4, they guard entrances and exits, openings of the digestive and urinary tract, as well as other openings to the body. And the 5th one is that they maintain body temperature. Okay? They do this by producing heat when the muscles are contracting to warm the body. One muscle will have one nerve that innervates it and then branches to each of the muscle fibers so they get their own supply. They will also have one artery that branches into capillaries for each of the muscle fibers. The muscle, again, is composed of connective tissue, blood vessels, and muscle fibers, and we'll start with the connective tissues. There are 3 layers of connective tissue on the muscle. Now, you want to imagine this sort of like the magic school bus, where we're going to go, that show where you go down deeper and deeper into the internal part of the muscle. So we're going to start externally on a large muscle, say your biceps brachii, and that whole muscle is going to be surrounded by the epimysium. This is a dense collagen fiber that separates the muscle from surrounding tissues and organs. It's connected to the deep fascia. Epi means above, and mysi is going to refer to muscle. So whenever we say mysi, you'll think of muscles. Now we're going to go deeper into the muscle. We're going to go past the epimysium around the whole muscle, and we're going to talk about the perimysium. The perimysium is the second level of membrane, and this is going to divide the muscle into a number of little compartments called fascicles. And each compartment is going to house a number of muscle fibers. Okay? Again, this is going to be made up of collagen, elastin. There'll be blood vessels and nerves for each fascicle, as it's branching from the larger blood vessels and nerves down to smaller to feed each fascicle. The term peri means around, and mysium refers to muscles. We're now looking at the 3rd layer of connective tissue. This is the innermost, called the endomysium, and this will surround the individual skeletal muscle fibers. And it will also connect other adjacent muscle fibers. Endo means inside, mys means muscle. Also found inside the endomysium, between that connective tissue and the muscle fiber itself, is going to be the capillary network. So there'll be small capillaries there for the individual muscle fiber. The nerve fibers are going to be trapped there so they can control the individual muscle fiber. And there will be another type of cell called a myosatellite cell. These are embryonic stem cells that gave rise to the muscle. They stay as individual cells, and they are going to function in the repair of the damaged muscle. Okay. Now we have a number of different fibers here. We have collagen fibers that are coming from the endomysium, the perimysium, and the epimysium. And these are all going to come together externally on the muscle itself, and they're going to form a bundle, and they're going to give rise to the tendon, which is a bundle, or if it's a broad, thin sheet of tendon-like material, it's called the aponeurosis. These will then attach the muscle to bone, directly or indirectly, and the collagen will extend directly into the bone matrix, and that will make it even a much stronger attachment. The second structure of the muscles themselves are the blood vessels and nerves. Now these are going to start on the epimysium as large vessels and large nerves. Then they're going to branch into the perimysium and then branch even farther into endomysium, and forming very small branches that will go to the individual muscle fibers to meet their needs: oxygen and nutrients from the blood vessels, nervous stimulation from the neuron. The third structure of the muscle is the muscle fiber cells themselves. Now, skeletal muscle fibers can be very long cells, up to 12 inches in length. They are multinucleate. This is because they start out as individual cells which then fuse, and each of the fused cells gives its nucleus to the large multinucleate cell, and they need these so they can produce enough enzymes and structural proteins for a very long cell to work. Muscle cells need a high amount of material because they have a high metabolic turnover, because they are doing continual removal and replacement of the organic molecules of the cell. Now, your muscle fibers, multinucleate, are made originally from myoblast cells. Blasts are builders. These are the embryonic stem cells of the muscle, and a group of the myoblasts will fuse together during embryonic growth, making the multinucleate fiber. A small handful of the myoblasts will stay unfused, and those unfused myoblasts will be called myosatellite cells, and these will live right underneath the endomysium and will be involved in help with injury and repair of any kind of skeletal muscle damage. This is just a diagram of a individual muscle fiber showing the striations that occur, the nuclei, the myofibrils, and it will also have numerous mitochondria within the cell because it needs a large amount of ATP. Alright, on our little journey in our magic school bus, we're now deep into the individual muscle fiber. Now, a muscle fiber, which is a cell, will have some general structures that a regular cell had that you learned about earlier in the semester, with some modifications. The plasma membrane of the muscle fiber is called the sarcolemma. The cytoplasm is called the sarcoplasm. Note, sarco means muscle. Okay. We have coming down from the surface of the cell, from the sarcolemma, going deep into the muscle tissue, what are little tubes called transverse tubules or T-tubules, which is more likely what I'll refer to them as. These are narrow tubes that are continuous with sarcolemma. They extend into the sarcoplasm. They're going to be filled with extracellular fluid, and they are going to be involved in transmitting an electrical impulse, known as an action potential, from the surface of the cell membrane down deep into the internal area of the muscle fiber. Okay. Now, think of these as forming passageways as sort of like a tunnel through a mountain, so they can take information in deeper, and this is going to allow for a quick communication to the muscle fibers for contraction. Okay. Myofibrils are located inside the muscle fiber. Now, the muscle fiber itself is going to be a long and tubular structure. Now think of it as being made up of a bunch of tiny, smaller, long tubular materials inside it. Those are called myofibrils. The fiber is large. The fibrils with the L are little. They're inside the muscle fiber. They're going to run the whole length of the muscle fiber, and those are going to be made up of myofilaments. Filaments are like fishing line. They're solid. The myofilaments are going to be protein filaments, and we're going to have two. We have a thin filament made of actin and a thick filament made of myosin. Now, there are, in one muscle fiber, hundreds to thousands of these myofibrils that are made up of many myofilaments. And the myofibrils are going to be responsible for the contraction. They're going to be anchored into the surface of the sarcolemma, and when they shorten, they pull on the surface, and that pulls the whole cell inward. And then the cell fiber, when it pulls, will ultimately, a number of these will extend to the tendons that will pull on the bone and result in movement. The next structure that we see in here is called the sarcoplasmic reticulum. This is a type of smooth endoplasmic reticulum, which, if you recall, I said have some specialized jobs in certain cells. This is one of them. This is going to be a calcium reservoir. It's going to store calcium and release calcium as needed, which is required for muscle contraction. Okay. Now the sarcoplasmic reticulum is going to surround a nice tubular network around each of the myofibrils, and then it will be tightly bound to it. And on either side of the T-tubule, where that's coming down internally to the cell, which surrounds the myofibrils, there is sarcoplasmic reticulum storage areas. They're larger areas, and they are called terminal cisternines. There's going to be 2 terminal cisternines with the T-tubule in the middle, and those together are called the triad, group of 3. Now, the sarcoplasmic reticulum is going to store a large amount of calcium in those 2 storage areas, the terminal cisternines. That's about 4,000 times as much calcium as found in the sarcoplasm itself. And the T-tubule will be in the middle to bring the electrical impulse down from the surface of the cell and cause the release of calcium. The next structure of the muscle fiber is found within the myofibrils. The myofibrils, the long tubes that are within, running the length of the muscle fiber, they're inside it. They are made up of repeating units of protein, and these repeating units are called sarcomeres. These are the functional units of the muscle itself. They're made up of myofilaments, which are the protein, actin, and myosin, and they are going to actually have within those little repeating units of the sarcomere an interaction between the myosin and the actin, which will cause that sarcomere to contract, and the next sarcomere will do the same thing, and the next one will do the same thing. So the whole myofibril units, the sarcomeres, will contract at the same time, causing the myofibril to contract, and then that will cause the whole muscle fiber to contract, and then many muscle fibers are doing this, causing the muscle itself to contract. Now, your sarcomere is made up of a number of different kinds of proteins. There's the thick filaments, which are myosin, the thin filaments, which are actin, and then there will be proteins that stabilize the position of the thick and thin filaments, because they have to be kept in the right spot so they can reach up and grab and pull each other in. And there's going to be proteins that regulate the interaction between the thick and thin filaments, when they can grab and when they can't grab each other, because you don't always want your muscles to be contracting. When you look at the sarcomere, it has a banded appearance, or in the sarcomere itself, you're going to have an A band, which is going to be dark in appearance, and then there will be an I band that straddles between 2 sarcomeres, which will have a light appearance. When we look at the slide, we saw the striations, the red and white banding pattern. The red stripes or striations were from the A band, and then the white or lighter ones were from the I band. So if you think of A bands as dark, that dark has the letter A in it. I bands as light, light has the letter I in it to help you remember it. And your A band is going to be made up of thick filaments, and your I is going to be made up of thin filaments only. Now within the A band we're going to have 3 subdivisions. Okay. The A band is going to run from one end of the myosin thick filament to the other end of the myosin thick filament. But within that we have some subdivisions. In the very middle there is a line of protein that goes all the way down, and that's called the M line, and this is going to help stabilize the position of the myosin. So think of middle and M as the same. Okay. Then on either side, including the M line, there is the H band. This is going to run from one side to the other where there is only myosin in this area, and then the remaining part of the A band, there will be 2 zones of overlap, one on either side, and these are areas where you're going to have both the myosin, the thick filament, and the actin, the thin filament, where they're overlapping with each other. The I band, which is the light band, contains thin filaments only. There should never be any thick filaments in there. And they are going to straddle between two different sarcomeres. They're going to run from the end of the A band and one sarcomere to the end of the A band in the next sarcomere. In the center of the I band, there is another layer of protein that forms the Z line or Z disc, as your lab book refers to it. Okay? And this is going to be the end of the sarcomere, and this is where we're going to have our actin, thin filaments, attached to the Z line. The Z lines are made up of proteins called actinins, okay? And thin filaments attached to this, and also the thick filaments will have an inner core of a protein called titin that will run to the Z line and hold it in place also. It kind of works like a spring. So when the Z line is pulled in, that core of titin will tighten up and form kind of a spring-like structure. Alright, this is just those same pictures we just saw, just all put together to kind of review the muscle structure from gross to microscopic anatomy. The muscle itself is made up of many bundles of muscle fibers called fascicles. The fascicle is made up of many muscle fibers, which are the cells. The muscle fibers are made up of myofibrils, which are inside the muscle fiber. Myofibrils are composed of myofilaments, and those myofilaments are made up of repeating units of thick and thin filaments called sarcomeres. The next 5 slides are just enlargements of figure 10.6 in the individual images, so you can see it up a little closer. This is the skeletal muscle itself with its epimysium surrounding the outside, the fascicles, which are surrounded by the perimysium, and the individual muscle fibers, which will be surrounded by the endomysium. Here's the muscle fascicle surrounded by the perimysium. You can see the branching blood vessels and nerves that came from the larger artery and nerve to feed the fascicle itself. And the individual muscle fibers are in here, surrounded by the endomysium. This is the figure of the muscle fiber itself with its endomysium, and you can see the nuclei, the myofibrils that are being pulled out here and individually and made up of the myofilaments. Here we are looking at one myofibril. Many make up a single muscle fiber, and you can see the sarcoplasmic reticulum with the enlarged areas called the terminal cisternines on either side of the yellow T-tubule. And you can also look and see a sarcomere, not well expanded, but a sarcomere. And now we have the individual sarcomere with the different layers in it. The A band, which is the thick filament and is dark in color, that is comprised of 3 zones, the M line in the middle. The H band, which is going to be the myosin on either side of the M line, where it is just myosin only, and the M line is also included in the H band. And then we have 2 zones of overlap where there is both the myosin and the actin. You can see part of the I band extending to the Z line, and the I band will extend to the next sarcomere and be part of the next sarcomere. That is a lighter band, so the I band is the light band, and they both have I in them. Okay, we're now going to go into more detail about the thick and thin filaments. We start with the thin filaments, which is a single thin filament will contain up to 4 proteins. It has 4 different types of proteins. They have different jobs. So it's not just made up of actin, but that's the majority of the protein. If you look at the figure, we have little balls. They look like little fishing bobbers or something. You make a nice chain of those little balls. The individual balls are called G-actin, which is globular actin. And you take and make a chain of the G-actins. You make a nice filament that is called F-actin, which stands for filamentous actin. And we're going to take 2 of these filaments and twist them around each other. Now, if you note on the little balls, they have a little yellow top. They look like there's a spot where the rest of it is red. They're yellow. That is the active site of the actin, where the myosin wants to grab it. Now we don't want them grabbing it all the time, so we're going to layer another protein across the active sites so that it covers the active sites from the myosin when we're not contracting. And that protein is called tropomyosin. And that's going to cover about 7 of the actin, the G-actins at one time. Now we don't want them it always covered. Sometimes we want to be able to move it off the active site so we can do contraction. So there is the 4th of the proteins called troponin, which sits on top of the tropomyosin and controls the 7-unit tropomyosin, and will pull it off the F-actin when there is a contraction. The trigger that is going to trigger it to move is going to be the presence of calcium, because troponin loves calcium. When there's no calcium, it'll stay in place, it'll hold the tropomyosin right in front of the F-actin and protect it. But when calcium comes along, it's going to reach for the calcium, move towards it, and pull the tropomyosin with it, and that then exposes the active sites to the myosin. The thick filament, the myosin, is just myosin within the thick filament, and the actual individual myosin molecules look like golf clubs. They have a nice tail which looks like the handle, a head that looks sort of like a golf club head, and in between the two you can see a little hinge, and this hinge works like a spring, so that when you pull the head back, it will cock it and it'll store that energy in the spring, so if given a chance it will then spring forward and release the energy. Now the myosin wants to reach up and grab the actin with its myosin heads. It cannot do that if the tropomyosin is covering the active sites. When the active sites are exposed by troponin, then the myosin can reach up and grab it. When it does reach up and grab the actin, this is called forming a cross bridge. And also located in the myosin is a protein called titin, which we mentioned earlier, that looks like a little spring, will extend to the Z-line to stabilize the position of the myosin. When the muscle fiber contracts, there will be changes within the sarcomere in those different bands that we mentioned earlier. When contraction occurs, the H zone and the I band will both get smaller. The zone of overlap will get larger. The Z lines will move closer together, but the A band will not change at all. The width will remain constant. And you can see the difference between the relaxed sarcomere and the contracted sarcomere in the figure. During the contraction, the thin filaments are going to be pulled inward by the thick filaments, the myosin head grabbing the active sites and pulling it inward. They're going to slide toward the center of each sarcomere. Contraction is going to stop when those I bands disappear and the Z lines are in contact with the ends of the filament. So you've pulled it in as far as you possibly can. Now, within one myofibril, every single sarcomere will be contracting by this sliding mechanism. And all the myofibrils within that one single muscle fiber will contract. Therefore, the muscle fiber gets shorter, and if a number of muscle fibers are doing it at the same time, that will determine the strength of the muscle contraction. This whole process of sliding the actin inward by the myosin is called the sliding filament theory of muscle contraction. For muscle contraction, there are a number of steps that have to be accomplished for this to occur. First, the muscle has to get excited, so the neuron that has to stimulate the skeletal muscle to contract. Okay. Second, after it gets excited and that action potential is going to travel on the sarcolemma, we have to somehow connect the excitation to the concept of a contraction, and that's called excitation-contraction coupling. And this is when the action potential is going to somehow stimulate the muscle contraction. The third thing is the actual contraction cycle itself, what's happening when the muscle is contracting. And 4 is relaxation, the stopping of the muscle contraction. The first step is excitation, nervous control of the skeletal muscle activity. The neuron is going to reach down and touch the skeletal muscle fiber at the neuromuscular junction. And neuron is a nerve cell, and it's going to be made up of a long axon coming from the cell body, and then it will branch, and at the end of the branches will be little rounded ends called synaptic terminals. And in each of these synaptic terminals, it's going to come in contact with the muscle fiber. It is filled with packets or vesicles of a neurotransmitter called acetylcholine. Neurotransmitters are going to be released from that synaptic vesicle, and they're going to make their way across to the muscle fiber and excite it. Okay. Some neurotransmitters, other types, will not excite it. Okay. Basically, the acetylcholine is going to change the permeability of the sarcolemma, the plasma membrane of the muscle fiber, and that's going to allow certain molecules to rush in there, sodium molecules, which will start the action potential. Now, looking at this figure, you can see where the muscle fiber and the neuron meet, the neuromuscular junction. Also, the part of the muscle fiber that is involved in the neuromuscular junction is called the motor end plate. And this is going to have a number of little receptors on it that are going to receive the incoming neurotransmitter, the acetylcholine. And when they sit on the receptors, they will open some gates and let sodium flow into the cell, which will trigger the production of an action potential on the sarcolemma. Okay. Also, you don't want to keep this going forever, so the acetylcholine is going to be quickly removed from the receptors and broken down by an enzyme called acetylcholine esterase, and then the acetylcholine is either broken down or is thrown away, or is going to be recycled back into vesicles in the synaptic terminal to be used again. We're going to go through the steps of what happens at that neuromuscular junction. In this picture you can see that we have the initial state where nothing is happening, but you can see the vesicles of acetylcholine in the synaptic terminal. You can see the muscle fiber, which is the tan structure, that has the little purple receptors on it, and they will respond to the binding of the acetylcholine and open those receptors. Located in the space between the neuron in the synaptic terminal and the muscle fiber itself, called the synaptic cleft, you can see those little half moons floating about, and those are your enzymes, acetylcholine esterase, ready to go to work to break down the acetylcholine when it is released. In this figure we can see that little lightning bolt, the red lightning bolt coming down, which is implying the action potential is arriving. Nothing has happened yet. It's just getting there, and it will begin the process of releasing the acetylcholine. The action potential has arrived. The vesicles of acetylcholine have fused with the plasma membrane, and through exocytosis are spitting out the red molecules of acetylcholine. They're now in the synaptic cleft, going to make their way across to the receptors on the motor end plate. Some of the acetylcholine has made their way over to the receptors. That's the little red ball of acetylcholine sitting on those little purple receptor gates. When the gates open, because acetylcholine has bound with them, that will trigger the influx of sodium, Na+, and the influx of sodium is going to be what triggers the action potential in the skeletal muscle. In the final step, the acetylcholine quickly moves in and then is pulled out of the receptors, and it binds with the acetylcholine esterase, which then either breaks it down to get rid of it or be used somewhere else in the body, or will recycle it back into vesicles in the synaptic terminal. This is all the steps of excitation put together on one slide, so you can view it once more in a continuous manner. The next step is going to be how excitation actually triggers the contraction. This is called excitation-contraction coupling. Once the excitation-contraction coupling is over, the contraction cycle is going to start, where it will then pull the actin in by the myosin and cause the sarcomere to actually shorten. Excitation-contraction coupling. When the action potential hits the sarcolemma, it runs along the sarcolemma, which is an excitable membrane, and then it goes down through the T-tubules. Remember that the T-tubules are part of the triad. On either side of the T-tubule is the enlarged part of the sarcoplasmic reticulum called the terminal cisternine, which is where most of the calcium is stored. When the electrical impulse or action potential runs down the T-tubule, it stimulates the terminal cisternines to release their calcium. Once the calcium is released into the cytosol, then the protein troponin, who likes calcium, sees it and binds with it, and when they bind with the calcium, it causes the troponin to move. When it moves, it also takes with it the tropomyosin that was blocking the active sites of the actin. Once the troponin-tropomyosin complex has moved off the active site of the actin, the myosin can now reach up and grab it and form the cross bridge, and the contraction cycle can start to begin. Once the muscle fiber starts contractions, it will increase the tension within the muscle fiber itself and increase the tension within the muscle, which is attached to the bone by the tendon, and the tendon will pull on the bone, and this will result in movement. The contraction cycle. This is going to begin with the arrival of the calcium into the sarcomere, which will then bind with the troponin. The calcium binds with the troponin, which causes it to shift its position, and the troponin now pulls the tropomyosin, which it is also bound to, off the active sites of the actin, exposing the active sites now to the heads of the myosin, which will now start reaching up and forming a cross bridge. Cross bridge formation is going to occur when the head of the myosin reaches up and grabs the active site of the actin. Now the head of the myosin has been activated and energized by using ATP. It breaks it down from ATP to ADP plus phosphate, releasing energy, which then gets stored in the little hinge. So now it's ready to snap back. So it'll reach up, grab it, and then snap back and pull the actin inward, the sliding filament theory. The myosin is also going to need to use ATP to release the actin and recock and get ready to do it again. Here the myosin head is pivoting, where it's using the energy that's stored in the hinge and snapping the head forward, pulling the actin inward. That energy came from ATP. It will pivot and keep pivoting and pulling the thin filaments inward towards the M line of the sarcomere. This is the sliding filament theory. When the head is done pivoting, it needs to detach from the actin using ATP and then storing that energy from the actin into the hinge when it recocks, so it can reach up and form another cross bridge and do it again. This is going to continue over and over again while it slides the thin actin filament inward towards the M line. The myosin head has recocked, has now stored the energy in the hinge, and is ready to reach up and grab the actin again, resulting in sliding it inward. Again, this is the sliding filament theory of muscle contraction. This is a review of the complete contraction cycle, starting from the calcium's arrival to remove the troponin and tropomyosin off the active heads, the myosin heads, which are activated with the energy reaching up and grabbing the actin, and then cocking the myosin head and recocking and pivoting it to bring it inward, then releasing, then recocking and grabbing again, so it slowly slides the actin inward towards the M line. Another review starting from the very beginning of the muscle contraction, where the acetylcholine is released and makes its way across the synaptic cleft to bind with the receptors, opening the gates, allowing the sodium in to start an action potential on the cell membrane. The action potential goes down the T tubules, which is surrounded by the sarcoplasmic reticulum, which is the calcium reservoir. The action potential triggers the release of the calcium. That will bind with the troponin, which moves the tropomyosin off the actin, and now the myosin can reach up and grab it, and it will slide the actin filaments inward, resulting in a contraction. The final step in the contraction is ending it. This is called the relaxation period after the muscle contraction stops. Number of things have to happen. First, the neuron has to stop the action potential production. Then the acetylcholine that was out in the synaptic space has to be broken down by acetylcholine esterase or picked back up so we can stop action potential generation on the muscle fiber. The muscle itself, the sarcoplasmic reticulum, has to reabsorb the calcium from the cytosol. That will allow the troponin to move the tropomyosin back over the active sites of the actin, and the contraction will then stop. Now with your muscle fibers, they'll start the relaxation period when the contraction has ended. This period will be done to return the muscle fiber to its original length prior to the contraction. Although contraction itself is an active process with a lot of pulling going on and a lot of energy being expended, your relaxation is not an active process. So we're going to depend on gravity and the contraction of opposing muscles to help return the sarcomere to its original length. The amount of tension that's produced by the individual muscle fiber is going to be dependent on the number of pivoting cross bridges that form. Your muscle fibers are either going to be on or off. You can't half stimulate it, so it's an all or none response to the stimulation. When the stimulated calcium ions are released, all the triads in the muscle fiber are releasing them. Therefore, all of the sarcomeres of the muscle fiber are responding to the contraction and contracting, so the whole fiber will contract. Tension production is going to be dependent on two things. The resting length of the sarcomere at the time of stimulation, which basically is that if the zone of overlap is too big or too small, it won't allow enough cross bridges to form, so it won't produce enough tension in the muscle fiber. The second thing that controls the amount of tension that is produced is the frequency of stimulation. This is how often is that muscle fiber stimulated. Is it stimulated once and then it just relaxes for a while before it gets hit another time, or is it stimulated repeatedly over a short time period? The frequency of stimulation is going to affect the internal concentration of calcium ions. And the more calcium ions you have that keep building up, if you keep getting stimulated quickly, that will make the contraction much stronger. The first type of frequency of stimulation that we're going to look at is called a twitch. The twitch is just a single contraction. It causes the muscle to contract completely, then relax completely, and then it goes into the relaxation phase for a while. That means the tension rises and then drops completely back to zero, so there's not enough tension to do any type of work. And the extent of time a twitch will last will depend on the different muscles, because some muscles, like your eye muscles, only have a couple of muscle fibers responding to a twitch, whereas your gastrocnemius or soleus in the calf muscles will have more fibers responding, and therefore they will have a twitch that's a little bit stronger and will last a little bit longer. The second type of stimulation is what's called trep. This is when the muscle fiber gets hit by another action potential at the very end of relaxation phase. As soon as it ends, it gets hit again, and then it makes a little bigger contraction. Then it gets hit again, and it hit again, and this will slowly build up the contraction to a maximum tension, and this is due to the fact that there is still some calcium left in the cytosol that can't be picked up completely between the two contractions. The third is wave summation. Now we're decreasing the amount of the relaxation period. We're hitting before we hit complete relaxation. So it's coming a little sooner, and this also is going to keep adding to the contraction's tension and keep building to a certain maximum where it will then stop. Okay. If wave summation continues, it will reach what's called incomplete tetanus. Incomplete tetanus is going to be the maximum amount of tension you can reach when you still have some degree of relaxation phase occurring. And so it's going to go up and down in like hills. You get hit, you rise, then you go down a little bit, then you rise, then you go down a little bit. Your wave summation and incomplete tetanus will result in the tension being about 4 times as much as we had accomplished in TREP. The last type of stimulation is called complete tetanus. In this case, we remove all relaxation phase. There is no time for that, so that there's just continual contraction, and it will reach a maximum contraction that will mean steady in tension and fairly high. Most of our muscle contractions do reach complete tetanus, such as standing or walking. Those are all incomplete tetanus for those to work properly. Tension produced by the whole muscle, this is the entire muscle, not the muscle fibers, is going to be determined by 2 things: the internal tension and external tension that's being produced by the muscles, the fibers that are being stimulated. Now your internal tension, that's the tension inside the cell, will build, and as it gets stronger, it will actually spill out to the connective tissues and the tendon. This will ultimately result in enough external tension that it will pull on the bone and cause movement. Your twitch does not produce enough internal tension to have it transferred to external tension. The second thing that's going to affect the amount of tension production by the whole muscle is how many muscle fibers are being stimulated at one time. Obviously, the more you stimulate, they're going to produce more internal tension, and therefore they'll produce more external tension. Your tension in the skeletal muscle is going to rise slowly, and you're going to do this in a slow, smooth way by controlling how many muscle fibers are going to be activated at one time. A motor unit is going to be a single motor neuron and all the muscle fibers it controls. Looking at the figure, we have 3 motor units, the purple, the red, and the blue, and you can see the neurons and then the muscle fibers that they're controlling. Okay. You have small motor units and then for fine control, and larger motor units for more gross control, larger movements. For contraction, we're going to do this gradually. First, we're going to start to get the small ones going. And if the small motor units are enough, then we'll just let them do the work. If they're not enough, we'll have to bring on more units, more and larger motor units, to produce more and more tension gradually until we can have the movement that we need. This process is called recruitment, where there is a smooth and steady increase in tension produced by increasing the active number of motor units. Okay. For peak tension, you have to have a maximum number of motor units in the muscle working. Okay. Now, this is great, but each motor unit can only last for so long. To maintain sustained complete tetanus, you have to have a number of motor units that work in a relay fashion. So motor unit red goes first, then when it's tired, blue picks up. When it's tired, the purple motor unit picks it up. That way you have a constant amount of tension, but each motor unit is working, then resting, and then they can be called back into play to take their turn when they are fully rested. Muscle tone is the resting tension in a muscle. Even though your muscle may not be working very much, it still has some motor units that are active. So this provides a little bit of tension. This is going to be involved in stabilizing the positions of your bones and joints, as well as your posture. And the greater the muscle tone, the higher the resting rate of metabolism. There are 2 types of contractions, isotonic and isometric. An isotonic contraction is what you think of when you're doing a muscle contraction, where the tension increases and the skeletal muscle length changes. So when you're walking or lifting things, you're doing isotonic contractions. An isometric contraction, and that means equal size, the tension is produced, but it's never enough to exceed the resistance of whatever it is you're trying to do, so the muscle doesn't change length. So if you're trying to push the wall down, you can't move it. You still can feel your muscles contracting, but they're not getting smaller. Energy use and muscular activity. An individual muscle has about 15 billion thick filaments. Each one will use 2,500 ATP per second during an active contraction. Unfortunately, we can't store ATP because it breaks down too fast, so we can't have a lot of it laying around. The resting muscle fiber will produce a small amount of excess ATP that it can use, but it will be used quickly once the muscle starts working. And basically it's just giving a little buffer so that the muscle can start increasing its production once contraction starts, so it gets the muscle contraction started, and then the remainder of the contraction, ATP production, will equal ATP use. During rest, the skeletal muscle is going to make more ATP than it's using. It can't store it as ATP because it breaks down, but we can store it as a different type of energy molecule. What ATP will do is transfer the energy in the form of a phosphate with that high energy bond to a molecule of creatine. This will form creatine phosphate, which is another type of high energy molecule, but this one can be stored. So your resting skeletal muscle fiber has about 6 times as much creatine phosphate as it does ATP for use later. At rest, the muscle will depend on aerobic metabolism fatty acids to produce ATP. They're going to absorb glucose while it's resting, but that absorbed glucose is going to be converted to glycogen and be stored for later use when they need to produce more ATP. Any extra ATP the resting muscle is making is going to be converted to creatine phosphate. So we're going to have stored in that resting muscle 2 different types of energy reserves. We've got the creatine phosphate, plus we have the glycogen in the muscle for when we start actively metabolizing. During moderate activity, the ATP is going to be made mainly from aerobic respiration of glucose. That glucose has been gotten from the stored glycogen, which we converted back to glucose. It can also use some fatty acids. The creatine phosphate that we had made during rest is quickly used up, and now ATP production is equal to ATP usage, all done from aerobic respiration. At peak activity, our ATP demands are enormous. Oxygen can't diffuse fast enough into the cell to meet our ATP demands, so we have to turn to fermentation. About 1/3 of the ATP will be produced by glycolysis, which is now making pyruvate faster than can be used because we don't have enough oxygen to send that into the citric acid cycle and use that pyruvate. The other 2/3 of ATP are going to be produced by fermentation. The leftover pyruvic acid, or that through fermentation, is going to be converted to lactic acid, and we have a buildup of hydrogen ions, which are going to make the cytosol very acidic, both of which are going to cause damage to the cell if we continue peak activity for any length of time. If you continue peak activity for any length of time, you will ultimately hit muscle fatigue, where it can no longer perform at the required level of activity. You're going to have used up your metabolic reserves. That drop in pH, where we produced all those hydrogens, is going to affect the muscle fibers and the muscle fibers' enzyme activity. We're going to have some damage, possibly, to the sarcolemma. The muscle fatigue is going to be cumulative. It's going to get more and more pronounced as more and more fibers are affected by it, and it will result basically in a gradual reduction in capabilities of the whole muscle. The recovery period for the muscle, where it can put all the conditions back to what they were like before it did the work, is going to involve getting rid of the lactic acid. Okay. Some of that will be converted back to pyruvic acid, where it can go into cellular respiration. Some can be converted back to glucose, where it can be stored as glycogen. We have an oxygen debt, so we need to get high oxygen consumption so we can restore our ATP, our creatine phosphate, and our glycogen concentrations. And we have to get rid of the heat that we produced when we were exercising and using muscle contraction, so that we can stabilize the body temperature and make sure the enzymes work correctly. Muscle fiber performance is going to depend on the type of muscle fibers in the muscle. We have 3 different types, and the physical conditioning of the muscle. The type of muscle fibers that you will have is going to be genetically predetermined. You have possibility of 3 fibers. You have fast fibers, which are white in color. They contract quickly, they fatigue quickly, and this is the majority of your muscle fibers. Now, for these to occur, to contract quickly and fatigue quickly, they have a very large diameter with a lot of myofibrils. They've got a large amount of glycogen reserve with a lot of enzymes to break down the glucose really quickly so that they can do the quick contractions. They don't have a lot of mitochondria because they're going to use up their ATP fast, and then they're fatigued, so they don't need to have an excessive amount because they're not going to do this for a long time. And most of the ATP is done through anaerobic production and glycolysis only, so it's pumping it out. And this will catabolize glucose only. This would be an example of this would be like your muscles if you were a weightlifter, where you do a big quick jerk up, hold the weight and then drop it fast, or a sprinter who runs quick and then they're exhausted at the end of the sprint. Slow fibers are red fibers. These are red in color due to the fact that they have more myoglobin in them. Myoglobin is a type of oxygen treasure chest. It will store extra oxygen, but it has a red coloration. So these fibers are going to have they're going to have called slow because they have a slow contraction. They do not fatigue easily, so they're just slow and steady. They have a small diameter, so the oxygen can diffuse quicker in there. This is going to allow them to do more cellular respiration. They have a large amount of mitochondria, so they have a lot of ATP production, doing a lot of cellular respiration. They have many capillaries, so they can bring in more oxygen supply. And they contain myoglobin. Again, this is an oxygen storage, so it will hold on to the oxygen and reserve it. So the hemoglobin will pick up oxygen, drop some off to the myoglobin, pick up more oxygen. So if you start your oxygen and the hemoglobin is starting to run out, it will go back to the myoglobin and put it back into service. An example of this would be the muscles of a marathon runner versus compared to the sprinter. The marathon runner has slow contractions. They contract for a long time. The third fiber is called an intermediate fiber, and this will have characteristics of both types. It'll have many capillaries and be slow to fatigue, like the slow fibers. It has little myoglobin for oxygen storage, like the fast fibers. And conditioning can convert some fast fibers into intermediate fibers and increase your stamina. Muscle hypertrophy means the enlargement of the muscle. Okay, this is going to be done by doing weightlifting. How this works is not by an increase in the number of muscle fibers, but by the diameter of each muscle fiber getting larger. You're going to have a little tearing of the sarcolemma, and then it will repair itself, so it allows the diameter to get bigger, and you're going to fill it up with more mitochondria, a larger glycogen reserve and glycolytic enzymes, more myofibrils, and more myofilaments. This allows your muscles to get bigger. Atrophy is the reduction of the muscle mass and strength, and this is due to a lack of use. So far, everything we've talked about has been pertaining to skeletal muscle fibers. Now we're going to look at some of the differences for cardiac muscle and smooth muscle. Cardiac muscle is involuntary striated muscle. The cells of cardiac muscle are called cardiocytes, and they are single nuclei cells that branch, and where they touch each other they are connected by intercalated discs. The intercalated discs are filled with gap junctions to allow for action potentials to be transmitted between the cells, as well as desmosomes that link the cells together but allow some flexing. The cells of cardiac muscles have short, broad T-tubules, but they don't have the terminal cisternees, and therefore they don't have a triad. They are dependent on aerobic metabolism only, so they have a lot of mitochondria and a lot of myoglobin, and they store glycogen and lipids for energy use. There are 4 special characteristics of the cardiac cells. that are different from the skeletal muscle cells. The first is automaticity, and this means the cells can contract without neural stimulation. So they just control the contractions themselves, which is by what's called the pacemaker cells. Now the pace and tension of the contraction can be adjusted by the nervous system, but the pacemaker cells set the initial rhythm. The third thing is that cell contraction lasts 10 times longer than that in the skeletal muscle, because the heart has to contract and squeeze those chambers to move the blood along. They can't have a rapid contraction. They need to stay contracted for a long time. And they can only do twitches. They contract completely, then they relax completely and rest, and then they contract again. They can't undergo wave summation or tetanus because it wouldn't be able to move the blood from one chamber to the next. Smooth muscle is involuntary non-striated muscles, and they are found in almost every organ, lining the respiratory, urinary tract, digestive system. They are made of spindle-shaped cells that have no myofibrils, no sarcomeres, and no T-tubules. The sarcoplasmic reticulum is throughout the sarcoplasm, and there's thick filaments scattered in the sarcoplasm, and the thin filaments are attached to dense bodies, which is a structure that is in the sarcoplasm on the cytoskeleton, and also adjacent cells can be bound together at the dense bodies, so the cells can pull on each other because they have no tendons. The main special characteristic of your smooth muscle, there are two of them, is plasticity, which is its ability to function over a large range of lengths. Now, if you look at our skeletal muscles, if they are extended too far, they will get injured and they won't work. So we have to control how far we extend and stretch the muscles. With the smooth muscles, they can stretch a large amount and still have the ability to contract even though they're stretched. Example would be in the digestive tract. Your stomach changes a large amount if you ate a big meal, but it still has to be able to contract to then move the food down. Or if you look at in a female, when they're pregnant, the uterus will increase 60 times its original size, but yet still has the ability for the smooth muscles to contract to get the baby out. The second characteristic is that they have special little cells called pace setter cells, which seem to set the rhythm of the smooth muscle contractions without any neural input.